Welcome to episode 48 of the Energy Balance Podcast, where we teach you how to live without constant hunger and cravings, fatigue, brain fog, poor sleep, and other low energy symptoms by maximizing your cellular energy. I'm Jay Feldman. I'm a health coach and independent health researcher, and joining me again today is my good friend Mike Fave. Mike and I have been studying health and nutrition together for a long time now, and Mike also draws on his experiences working within the healthcare industry. Today's episode will be part 2 of our series discussing the Croissant diet, and in part 1 we started digging into the reactive oxygen species theory of obesity, where we discussed the major issues with this theory and dug into the biochemistry and physiology a little bit, and today we'll be continuing that by discussing the SCD1 theory of obesity, which builds on the reactive oxygen species theory of obesity. And the SCD1 theory basically is the idea that elevated levels of SCD1 cause obesity, and that decreasing SCD1 by excessively increasing oxidative stress is the best way to lose fat. So we'll be discussing the major issues there, as well as why fructose does not drive fat gain or necessarily increase SCD1. We'll be talking about why the polyunsaturated fats are harmful even if they decrease SCD1, and we'll again be discussing why fat burning is not the answer for healthy fat loss. If you are new to this podcast, then after listening through today's episode, I'd highly recommend you go back and listen through episodes 1 through 7, where we took some time to create a foundation as far as the bioenergetic view of health is concerned. To check out the show notes for today's episode, head over to jayfeldmanwellness.com/podcast, where you'll find the articles and studies and anything else that we reference throughout today's episode. And if you are struggling with any low energy symptoms, whether that is weight gain or chronic cravings and hunger, fatigue, joint pain, gut or digestive issues, brain fog, poor sleep or hormonal imbalances, or any other low energy symptoms or chronic health conditions, then head over to jayfeldmanwellness.com/energy, where you can sign up for a free energy balance mini course, where I will walk you through the main things that you want to do as far as diet and lifestyle are concerned, so that you can maximize your cellular energy. And I'll also explain why this is the key to resolving these symptoms and conditions. So to sign up for that free energy balance mini course, head over to jayfeldmanwellness.com/energy. And with that, let's get started. So the second major component of Brad's diet perspective, or perspective as far as the best way to achieve weight loss and health, has to do with his SCD1 theory of obesity. So I figured we would just start by explaining what SCD1 is and like where it's involved and when we're going to see elevations and decreases, because that's a huge part of what he's seeing as a major factor in terms of obesity and health. So SCD1 stands for steroyl-CoA desaturase 1, which is, it sounds, I guess, a little complicated, but it does something that's relatively simple, which is that it converts saturated fats into monounsaturated fats. And it tends to be very particular saturated fats, which are the ones that we are producing ourselves. And so SCD is found mostly in the liver and in body fat, which is where you would see fat being produced typically. And when there is excess amounts of fat being produced, you see an increase in activity of SCD1, basically converting that saturated fat, which we only produce saturated fat immediately, and then we convert that saturated fat to a monounsaturated fat. And as we've talked about before, both saturated fats and monounsaturated fats are extremely stable, especially when you compare them to polyunsaturated fats. So while a monounsaturated fat might be slightly less stable than a saturated fat, it's still an extremely stable fat that does have various purposes in our bodies, and we'll get to that and why it's important. But basically, so when you'll see an increase in SCD1 is when you are producing an excessive amount of fat, and so that would be a state called lipogenesis, where you're producing fat. That's what lipogenesis means. And along with that, there tends to be excessive amounts of inflammation too. And so some of the times when you might see this, and we'll dig into this a little bit more, is For example, in like a rat, when they're fed excessive amounts of fructose and they've got a lot of endotoxin circulating, or they're also eating a lot of polyunsaturated fats, you'll see a lot of lipogenesis. You'll see it in states of obesity and diabetes or non-alcoholic fatty liver disease. You'll see it in basically a ton of different excessively stressful states, and states where you have basically a blockage of energy production, a blockage of the ability to convert some substrate that's coming from food into energy, which basically leads to that substrate being diverted toward the production of fat. So in the liver, this is where we'll be talking about this the most, but basically an example was for the rat situation, where if you have a lot of polyunsaturated fats or you have a lot of endotoxin, that's going to block the ability to convert that fructose to energy, and instead that fructose basically leads to a buildup of acetyl-CoA, because you have blockages along the electron transport chain and blockages along the Krebs cycle that lead to a buildup of acetyl-CoA, and that acetyl-CoA gets shunted off and diverted to a pathway that leads to fat production. So you end up with a lot of fat being produced, and then you have an adaptive increase in SCD1 to convert some of that fat to monounsaturated fat so it can serve various purposes.
Yeah, I also think it's important to note here that in obesity, diabetes, non-alcoholic fatty liver disease, excess fructose feeding, and most of these metabolic syndrome states, which is like a whole classification of diseases that have like sort of the same features with hyperlipidemia, they're generally characterized by states of high endotoxin, or they call it low-grade endotoxemia in the blood. And even in other inflammatory states, there's like a generic response in the serum or in the blood. It's called an acute phase response, and it's categorized by the liver producing a series of proteins, but in response to inflammatory mediators like tumor necrosis factor alpha, and then also upregulating lipogenesis, and that can increase cholesterol and increase triglycerides in the bloodstream, because they have immune modulating effects or immunoprotective effects, and then they help deal with inflammation and toxins and basically it's just a huge general response to stress or inflammation. And so basically, when you have studies where you're going to feed rats a ton of fructose, what they've already shown, and this happens in humans, happens in monkeys, happens in rats, happens in mice, that you find high levels of endotoxin in the portal vein, because fructose isn't absorbed very well by the mammals studied, or these types of mammals studied without glucose. So basically what you're seeing is a state of endotoxemia. So when you're trying to draw parallels there, then you can also see that in with the obesity and diabetes, et cetera. They're all characterized by this high amount of endotoxin. And so so with the acute phase response, when you create hyperlipidemia, where you have that elevation in triglycerides and cholesterol, that is because the liver is upregulating fatty acid production. And when you do upregulate fatty acid production in these states, it's going to be saturated. So SCD1 also upregulates as well to convert that saturated fat into, or some of that saturated fat into monounsaturated fat. So like it's sort of an association to just say, oh, you see SCD1 in these in obesity, diabetes, and all these rats fed fructose. So therefore, sugar or fructose or whatever is causing this upregulation SCD1, and an SCD1 is what's causing fat gain. There's much more to the picture than that. It's not just a straight pathway. You have to consider the other context, which is that the inflammatory states are upregulating the acute phase response as well.
Yeah. And we'll dig into all that in more detail. But as you said, one of the more common things that will drive that state is endotoxin, and that will be a huge confounding factor that we'll discuss. And so basically what we're getting at is that SCD1 is a part of an adaptive response to an inflammatory state that involves a lot of fat production. As you said, endotoxin can be involved there because fat production plays protective effects in our immune system, but also fat production is something that's going to happen anytime we have excess substrate and we can't convert it to energy. And again, that fat does have various other uses too, just in our regular health of basically all sorts of organs and tissues, like our skin and eyes in particular are noteworthy ones where you have various cholesterol or lipid components that are extremely necessary. And so SCD1 is, again, it's part of this kind of adaptive cascade. And it also has some other effects that are noteworthy. So for one is that when you so one thing that it does is it actually helps to slow down mitochondrial respiration. And as we always talk about, that's generally not a good thing. But it also makes sense as an adaptive situation, because when you are producing excessive amounts of fat, that's, as we said, a state of inflammation, a state of blocked respiration. And so you don't want to be further driving that respiration. We talked about this a lot in the last episode, where part one of the series, where when you're producing energy very inefficiently, and you're producing a lot of oxidative compounds, a lot of reactive oxygen species, and causing a lot of oxidative stress, that can be extremely damaging to the cellular structure, all the nucleic acids in there, the protein, and everything. It can damage the cell considerably and lead to death. So when that's happening, there are adaptive responses that are saying, hey, let's slow down there. Like, we don't need to keep pushing this really inefficient energy production. And so SCD1 is one factor there that will help to do that, because if there's a lot of fat being produced from excess substrate like glucose or fructose in a stressed out, inflamed liver, then you don't want to keep trying to force that stressed, inflamed cell, or those cells and those tissues, to keep trying to produce energy. It's going to cause a lot of damage. So
it's basically a release valve.
Exactly. Yeah. And so that's one effect of SCD1 that's noteworthy, and kind of has this adaptive protective effect beyond just kind of repurposing some of the fats that are produced. And so, as we said, elevated amounts of SCD1 are seen in basically degenerative states that involve metabolic syndrome. So insulin resistance and diabetes are major factors there. Stress is a major factor there. And so it is noteworthy that along with just, it's not necessarily just lipogenesis, but also oxidative stress. Those tend to go hand in hand. That is going to be driving SCD1. And that's going to, of course, play a major role, or it does play a major role in Brad's perspective here, because he tends to see that production of reactive oxygen species as a good thing. And so we'll delve into how that relates to SCD1. But along with that too, there are certain scenarios where excessive amounts of oxidative stress, very high levels of reactive oxygen species, will lower SCD1. And that's kind of where he sees fatty acid oxidation as a good thing. And we'll get to that. But,
which is, questionable.
Right. Definitely questionable. So, I guess.
Before you go forward with getting to that, I just want to point out here that the parallel where if you have a high amount of substrate, so say if you're eating a high carb diet, like a large amount of carbs, then you can get some fat production in the liver. That is not the same thing as elevating and the acute phase response from like something like inflammation induced by endotoxin. While they can both, even at least in the rats, they can both have this upregulation of SCD1. So both states don't necessarily mean pathology. Whereas one state is a pathology and the other state is just, that's physiologic. That's what you would want to happen. If you're eating it, at least, especially in rats or mice, if you're gonna feed them a very high sugar diet, you're gonna see their livers produce a lot of fat, and that's just normal. So you have to look at the whole context here, and the other thing is you have to. With that said, because this applies to things like physiologic insulin resistance versus pathologic insulin resistance or things like that, it really depends on the context. So you can't just be sweeping generalization of, SCD1's upregulated. Well, it's like, when the liver is going to produce a large amount of fat from substrate or in an acute phase response or from an inflammatory response, whatever, then yes, SCD1, especially in rats and mice, will be upregulated. I think that's important to point out, to make that distinction so that it's not just across the board, this is bad.
Right. And as we'll get to too, I know you mentioned like having some sugar or fructose tending to increase lipogenesis, and it does, but in very varying amounts depending on the organism, depending on what kind of state they're in, what else is present, like polyunsaturated fats and endotoxin. So we'll get to that. But it is worth bringing up that, again, it very similarly to the situation with reactive oxygen species and hormesis and all of those quote unquote stress pathways, is it depends on the context through which these things are being increased or we're seeing high levels. And just because they're, in this case, just because it's present during pathology or in pathological states, doesn't mean that any increases are inherently problematic. However, and again, they are adaptive, so it's worth mentioning that. However, I do think that generally we don't want to see high SCD1, but we'll get there. So,
but there's there's difference in when it's occurring in general. I think that's important because so we don't just have like, anytime you see it, because I think that's sort of what the narrative is or implies so far, for at least from this perspective. I guess you could call it Brad's perspective or the fire in the bottle perspective, whatever.
Yeah. So let's explain that. So we kind of mentioned what SCD1 is, but the perspective from that Brad is putting forth through the fire in a bottle perspective is that basically because you see SCD1 increasing in all these situations, and because SCD1 increases the unsaturation of saturated fat from fully saturated fat to monounsaturated fat, we don't want to be seeing high levels of SCD1, which, as we said, definitely makes sense to an extent. So we'll get there. But so basically, from his view, decreasing SCD1 is basically paramount. And if you don't, and you have a lot of monounsaturated fat production, that's not going to lead to as much reactive oxygen species production as saturated fats. And that's going to prevent all those effects that he's saying are good from excessive oxidative stress. So we talked about this in the last one, where from his view, you want to see excessive amounts of reactive oxygen species being produced through the oxidation of mostly saturated fats. And that's going to lead to all of those defensive reactions, the uncoupling and the activation of all those kind of stress pathways, and eventually fat loss. And so he's saying if you have all these monounsaturated fats from the upregulation of SCD1, then you're going to have a problem. You're not going to be able to produce enough reactive oxygen species, and then you're going to be left without being able to burn fat and without this fat browning state and without this thermogenic state.
And uncoupling and all that, yeah.
Right.
Which are all adaptive mechanisms. They're all to deal with the issue of too much ROS.
Right. Yeah, so already I don't see that as, I see, we already acknowledge that we see less oxidative stress as a good thing, but from his perspective, that's not a good thing, and that's why you want to decrease SCD1 as much as possible. And so he's got a quote talking about this where he was saying that Native Americans they got fat if they ate only maple syrup. I think they become fat for the same reason that mice fed a high sugar, very low fat diet did. Massive upregulations of SCD1 and lipogenesis in their livers, which then exported huge quantities of monounsaturated fatty acids. These Native Americans would have been pathologically unable to trigger sufficient ROS production in their mitochondria when burning fat to induce uncoupling protein-mediated thermogenesis.
I just think it's funny that it's saying it's the pathology is that they're unable to trigger sufficient ROS, but it's like your cell is recognizing it as a pathology because it's moving towards uncoupling to deal with the excess ROS.
Right, right. Where you're saying, so just to clarify, like you're saying that in the state of reactive, like if you have a lot of oxidative stress, the cell sees that as a problem, and so it leads to uncoupling, and so that seems pathological to us. Of course, we know that Brad is coming from a perspective where that's not pathological. That's supposedly ideal. We discussed why we don't agree with that last time. Yeah. But it is, I find it funny too.
I think the fact that the cell is having an adaptive response to it, would at least to me seems to indicate some type of pathology on the cellular level.
Yeah. I would generally agree. Although I would generally agree. I mean, these pathways I definitely see as adaptations towards stress. And as we've kind of clarified before, I think adaptation can be really beneficial if it's adaptation to increased energy production. And it can sometimes lead to uncoupling also. It can sometimes lead to autophagy or mitochondrial biogenesis. So those are
it's always context.
Right.
I know we always say that and it's annoying, but it is always context.
Right. So anyway, I think with that quote, I mean, there's some things that we'll dig into there, but again, the main thing is that he's saying upregulation of SCD1 due to lipogenesis in the liver causes a lot of monounsaturated fat production, which is unable to produce enough oxidative stress. And that leads to a lack of uncoupling and a lack of thermogenesis.
So on the flip side here, though, if you So just, I guess, to paint the picture, on the flip side, if you had a high amount of SCD1 and you had increased amount of monounsaturated fatty acids being produced with the SCD1, then the fat cells would basically not generate as much ROS, and they would be insulin sensitive, so that basically any carbohydrate that you would eat would be shuttled into the fat cell with insulin and then be converted into fats and stored as fats. And so basically that's like the opposite of what he's saying. Or that's what he's saying essentially, is if you have too much SCD1, too much MUFA or polyunsaturated fatty acids within the fat cell, then they would be insulin sensitive. And if they are insulin sensitive, then they can take in glucose and convert it and store it as fat. And so the goal here is to create insulin insensitivity or insulin resistance, so that the fat cells are unable to take in glucose essentially, or that's glucose, and basically store it as fat. And then they'd also upregulate their burning of fat.
Right.
And that's because with the increased ROS, and when you get the increased uncoupling protein production, then basically the electron transport chain and the Krebs cycle aren't linked together anymore. So you're just burning the fats away as heat.
Right.
So that's what he's saying you want to do. I guess that last part that I just discussed, whereas the previous part is what you don't want to do. You don't want the fat cells to be insulin sensitive. And then I think in the last podcast, I think we talked about at one point the spectrum of fatty acids where you have, or glucose and fatty acids from an insulin sensitivity point of view, where when you have a lot of carbohydrate, you increase insulin sensitivity. And then on the opposite end, you have a lot of saturated fatty acids, which would decrease insulin sensitivity. And this is purely from a burning perspective, where polyunsaturated fatty acids are closer to glucose and monounsaturated fatty acids are closer to saturated fatty acids, but not quite as strong at promoting insulin resistance as the saturated fatty acids.
Right. Yeah. Yeah. And so with this is part of why Brad is not a fan of fructose. And that's because fructose, especially in these studies looking at mice and rats, which we'll dig into, fructose increases SCD1. And he argues that this is at least in part because it's producing a much lower level of reactive oxygen species than fatty acid oxidation. This is also why he thinks that insulin is a problem, because you see increases in insulin in all these situations where you're seeing increases in SCD1. And then along with this as well is that he's saying that fatty acid oxidation is able to depress SCD1. He's saying that I should specify saturated fatty acid oxidation. So what he's suggesting is that if you're oxidizing only saturated fats, you're going to produce extremely high levels of oxidative stress of reactive oxygen species, and that's going to actually depress SCD1, and that's also going to allow fatty acid oxidation to continue, since high levels of SCD1 or increases in SCD1 will decrease mitochondrial respiration. They'll decrease fat oxidation, as we talked about, because basically when you're producing fat, you don't also want to be burning fat. They're kind of opposing pathways. So this is kind of, again, support for his view that you want to be continuing to increase fatty acid oxidation from saturated fats. In terms of SCD1, it will help to decrease it. And we'll talk about why we don't necessarily agree with that, but that's just kind of laying out some of his most important views there, I would say, or most central views as far as SCD1 goes.
Okay.
So Brad also talks about the relationship between polyunsaturated fats and SCD1, where the polyunsaturated fats tend to actually decrease SCD1, which kind of has juxtaposing effects, considering his other opinions on on polyunsaturated fats, where they cause less oxidative stress when they're oxidized in the mitochondria. So because of that, they're not as ideal from that standpoint, but they do more directly decrease SCD1, and that seems to be more of a regulatory mechanism, potentially because there are already unsaturated fats present. So SCD1 doesn't need to produce as many of the unsaturated fats, but may also be kind of more of a regulatory situation where the polyunsaturated fats tend to decrease the output of fat or exportation of fat.
And production of fat by the liver, which is something we've talked about before.
Right.
And that's through, I think the mechanism was that we mentioned before. The study was showing that with increased polyunsaturated fatty acids, I think it increased ER stress inside the cell. and then it wasn't able to properly produce fatty acids to export, which I think we talked about being a bad thing in the context of generating an acute phase response.
Right. Right. Basically preventing the adaptive response.
Yep.
So while they might decrease SCD1, we don't necessarily see that as a good thing. But Brad does mention that part as a good thing, and he says that there are certain polyunsaturated fats that their decrease in SCD1 outweighs the lack of oxidative stress that they would cause, but others that might not be the case. So we'll talk about that more too. But I just wanted to mention that as one part of his kind of position, and then another as well, which is that he's recommending other things to decrease SCD1 that all involve similar stress pathways as far as the activation of AMP kinase and MAP kinase, MAPK. And so that would involve things like metformin that he mentions and berberine, which is kind of looked at as an herbal form of metformin. And we've talked about this before as far as how those things are basically directly inhibit the production of energy at the electron transport chain, which is what leads to a lack of energy and leads to the increase in activity of AMP kinase and the activity of all these stress cascades and the wasting of sugar through glycolysis and the increased production of lactate because of that. So it basically causes this kind of disastrous state, which, yes, increases AMPK, yes, may increase uncoupling, yes, may decrease SCD1, but definitely wouldn't look at it as a good thing. But I guess we'll get there in a little more detail in a bit.
Yeah. And then also, I mean, the other thing with the metformin was its inhibition on the Cori cycle, which can affect everything going on in the liver as well by depleting ATP to basically convert that lactate back to glucose. It will inhibit that whole process.
Yeah, so you end up being stuck with a huge accumulation of lactate.
Yeah. And the liver can't do anything with it, and the cells are just like, I mean, a known side effect with a decent amount of mortality of metformin is metformin lactic acidosis, where if metformin reaches high enough concentrations, you essentially, but I mean, and that, I'm not saying that because you should never take metformin. I'm not telling anybody that, but what I'm saying is that that's how metformin's mechanism of action is basically to push towards glycolysis and essentially waste your substrate. And so, yes, it will lower blood sugar levels that way, but it doesn't push it towards things, toward the substrate towards oxidizing through cell respiration.
Right. And it does actually, just as a side note, and I think we've mentioned this before, but it also has some beneficial effects on the gut, which
as an antibiotic, yeah.
Right. Which is similar to berberine and similar to a lot of kind of phytochemicals or flavonoids and things from plant compounds that when they're in the gut, they have beneficial effects. And when they're absorbed systemically due to high doses, they have relatively toxic effects that people consider to be antioxidant inducing and beneficial, but they're doing that through stress. And again, that tends to be the certain types of those chemicals or compounds in like in foods, especially from like or herbs, but specifically tends to be from like the leaves and seeds, especially since those are relatively toxic to microbes, but also toxic to us if we're absorbing them systemically. So,
yeah. And I think you can see this too with like the effects of either like high fat, high sugar feeding, which is how they would produce like a fatty liver and upregulate SCD1, or even just like a high fructose feeding. The effects in like a germ-free mice versus a non-germ-free mice are pretty different as far as what happens metabolically. And I think you also get SCD1 upregulation in those states. But as far as like pathology and whatnot, it's not there because you don't have the high amount of endotoxin coming from the gut, especially in like a high fructose feeding model. So there's like, that is a very big moderator of, you have to keep that in mind for any drug or for a lot of the feeding trials or metabolic studies.
Mm-hmm. Yeah. And I mean, that's kind of a nice transition. Why don't we start talking about some of the problems, which we've kind of touched on along the way, but let's really dig into some of the problems with these perspectives that all we want to do is just decrease SCD1. Anything that decreases SCD1 is a good thing. We want to be producing a ton of reactive oxygen species to help decrease SCD1. Things that, and the way to do that is through fatty acid oxidation. And the way not to do that is with fructose and insulin. So, yeah, why don't we dig into all that?
I guess you might want to start because this was your favorite point here, where you basically fatty acid oxidation couldn't produce I mean, at least based on the study that I think this is the study that Brad was actually using, is I don't think fatty acid oxidation could produce as much ROS as that was needed to actually shut down SCD1. And I was just looking at comparing it. In the study, they compared it with different levels of hydrogen peroxide at the cellular level to see like how much was needed to actually shut down SCD1. And essentially it was more than was needed for like obesity and diabetes, which are states characterized by high ROS at the cellular level. So it's essentially calling for maintaining high levels of ROS at the cellular, or maintaining high levels of ROS in general, more than states of obesity and diabetes, to shut down SCD1 production, so that so that you don't produce monounsaturated fatty acids. You can keep your fat cells insulin resistant and continue to uncouple and burn through your fatty acid stores.
Yeah, it's basically like an insane emergency situation. But yeah, so to add some context there, basically this idea that glucose oxidation is going to produce some amount of reactive oxygen species, and that's increasing SCD1. And then fat oxidation is going to produce way more, and that's going to decrease SCD1, and that kind of excessively high oxidative stress, I think, is very unlikely, especially when you're looking at the study. And so what they basically did is they introduced all sorts of different damaging things to the liver that lead to a defensive reaction of lipogenesis, and that leads to all this inflammation. They used endotoxin in a couple of different doses, which we talked about quite a bit. They also used other factors like angiotensin, norepinephrine. They exposed them to hypoxia, and then also sugar, and we'll get
in
to
high
that.
amounts. Yeah,
right. And so what they found was that basically, in response to like things like endotoxin and hypoxia and norepinephrine and angiotensin, all of these stress factors, they all increased SCD1. And again, that makes sense. As we talked about earlier, SCD1 is there as kind of one of those adaptive stress pathways to, when there's a lot of lipogenesis going on, which is a part of that defensive reaction in the liver. And so basically, if you're arguing, the first argument here is that fatty acid oxidation is going to produce more reactive oxygen species than these other things. So you're saying that basically a pretty decent dose of endotoxin, and when we were looking at this dose, it was insanely high compared to like endotoxin.
And it was on an intracellular level too, right?
Yeah.
But what was it like? It was what, 50 nanograms or something? Mm-hmm. And when you're looking at, I think what we looked at was, if you looked at like a gram negative infection, you had blood plasma levels of, what was it? It
was in the pico. It was like up to 500 picograms.
Yeah, it was
right?
like 450 picograms. So like we're on a completely different scale. Like we're at completely different units by what, like a factor of 1,000.
Right. Yeah. I mean, 500 picograms would be half of a nanogram. So, which is 1/100th of what they were using, the dose that they were using for endotoxin. So they used 100 times that dose in the cell directly, and found that produced a lot of oxidative stress, and that oxidative stress increased SCD1. And so what Brad is basically arguing is that forget endotoxin, forget all those other things. If you're just burning saturated fats, you're going to produce even more oxidative stress than that, which I think is I mean, there's definitely nothing in that study to suggest that that's the case.
Well, the other thing too is that that's not accounting for the cell upregulating uncoupling or anything like that to decrease ROS production as well. And it's obvious that the cell doesn't want that much ROS. That's why it is undergoing these adaptive mechanisms like increasing brown fat or anything like that. But to say that that is the best way to lose body weight, I think is, or I don't know if he said it was the best way. I don't want to like make a straw man argument for him, but like essentially trying to lose body fat through that way, I think is questionable. Like, what are you exchanging? Like, if you're trying to, first of all, we're not reaching that much ROS, but what's the trade-off of trying to get that much ROS to induce uncoupling and upregulation of brown fat and cause physiologic insulin resistance and at these ridiculous levels or whatever is being proposed?
Yeah. And so along with this too is all of these stress-inducing factors tend to increase fatty acid oxidation. Generally, anything that increases stress does that. So when you're saying that something like endotoxin or hypoxia increases reactive oxygen species considerably and increases SCD1 and increases fatty acid oxidation. If that fatty acid oxidation was able to produce enough reactive oxygen species to decrease SCD1, then those other things wouldn't be able to increase SCD1. Then after endotoxin, you would see a decrease in SCD1. You wouldn't see an increase. So those things are pretty hard to reconcile. Yeah. I don't see how you can have both of those things. And another was norepinephrine, which again, notoriously increases
fat oxidation and lipolysis or lipolysis.
Yeah. So if you have a lot of norepinephrine increasing fatty acid oxidation and increasing SCD1, it's hard to argue that increasing fatty acid oxidation decreases SCD1.
That's as long as your tissues are saturated with, from this perspective, saturated with saturated fats.
Right.
If you have high amount of PUFA or monounsaturated fats in your fat stores, then when you increase norepinephrine, or even on the cellular level, when you increase norepinephrine, then technically you would still be insulin sensitive if you were going to oxidize them.
Sure. Right. Yeah. And along with that too, as you mentioned, like as we were talking about in the study, they were looking at obesity states and diabetic states, which are states of insulin resistance. So like in talking about this, they are in an insulin resistant state.
And they already have elevated free fatty acids.
Yeah.
They are oxidizing. Those states are, especially diabetes type 2, is characterized by the oxidation of fatty acid in high states of insulin and blood sugar levels. It is literally an inability to oxidize glucose.
Yeah.
At least appropriately.
Yeah. And in that state that they're looking at, there is increases in SCD1, not decreases. So just looking at all of these things, I don't see a way that you can argue that fatty acid oxidation is going to produce so much oxidative stress that it's going to decrease SCD1. And as we get to as well, like the next kind of point there is that if you're getting to that point where you're producing so much oxidative stress that you're decreasing SCD1, things are pretty bad. Like it's a really degenerative state. So I'm going to pull up this.
It's the perfect time to pull it up.
I'm going to pull up this figure from that study where they're looking at, on one hand, we have the adaptive stage, which is where you're in an obesity, diabetes type state, and you're producing a decent amount of oxidative stress here. The glucose is not being used efficiently. You are going to be driving towards fatty acid oxidation and whatever else. But in this case, because you're in the liver and you're not converting that sugar to energy, you're actually going to be producing a lot of fat, which is what leads to, that's the lipogenic state. And that leads to the increases in SCD1 as a, backup mechanism or an adaptive mechanism, converting those saturated fats to monounsaturated fats. So we see that on the left where you have, in this non-ideal state, you have an increase in SCD1. It's blocking fatty acid oxidation. Which also happens, it's also blocking pathways that lead to apoptosis. And, you have this kind of first stage. And then on the right.
Before you get there, I think it's important to show here that you're upregulating saturated fatty acid production in obesity and diabetes. And then SCD1 is upregulating concurrently with the saturated fatty acid production and converting those saturated fatty acids into monounsaturated fatty acids. So I think it's you produce palmitic and stearic acid, and then it converts it to palmitoleic and oleic acid. And then it basically pushes them into triglycerides where the liver can push them out into the bloodstream and move them towards the cells and tissues and what have you. So that's the first pathway on the left. And then in that state, you also have an increase in glucose and insulin as well.
Yeah. Pathologically, if you want to call it that, because they aren't, glucose isn't being used well and you have excess
high
insulin.
Amounts of fatty acids in the bloodstream. And then your body's trying to increase. And so the glucose can be upregulated from gluconeogenesis at this point as well, and also from intake. And then insulin is upregulating as well, trying to get that glucose into the cell. So you sort of have this like mishmash of substrate in the bloodstream at this point with high amounts of fatty acids and then also glucose and insulin. And basically the first diagram is just showing that the SCD1 here is actually adaptive because it's converting all the saturated fatty acids not all of them, but converting some of the saturated fatty acids being produced into monounsaturated fatty acids to be exported. And this is important because and there's a study that I guess you can put in the show notes, but without SCD1, without high levels of SCD1, you'll upregulate saturated fatty acid production. And then you can essentially it'll just stay in the liver and you just have really high amounts of you get fatty liver essentially. But then you can go into the maladaptive one next, which is you got it. Yeah.
Yeah. Well, and just to clarify too, when you're the problem in that state where you're mentioning the fatty liver and you're not able to convert those saturated fats to monounsaturated fats, like that is an it's worth noting that that's an adaptive process. Also, we don't even want to be in a position where we're producing a ton of fat in the liver in the first place. That is already a sign of things like obesity and diabetes. Of course, it can happen outside of those stages and in low amounts, but in large amounts, it shouldn't really be happening ideally.
Yeah.
So we have this not great situation on the left: obesity, diabetes, all these problems, and high SCD1. On the right, you have what they say is the maladaptive stage, where you have so much oxidative stress, things have gone so wrong that you have an inhibition of SCD1, and at that point, you no longer have an inhibition of fatty acid oxidation. You also no longer have an inhibition of apoptosis, and this is a state that they see in lipotoxic cardiomyopathy. So basically, in heart damage. So it's
this was done with cardiomyocytes. So this was looking at the cells specifically, and what happens when excess nutrient stimuli or humoral stimuli, and so that's basically with, I think, the norepinephrine, lipopolysaccharide, angiotensin, endothelin 1 hypoxia, or if you have like exposed to high amounts of glucose, you had the increased saturated fatty acids. That's basically the context of the picture. And then when you're going on the maladaptive side, the only differentiator between them is on the maladaptive side, you had such high amounts of strong oxidative stress that it actually turned off SCD1. And then you just basically had a high amount of saturated fatty acids at the cellular level that led to increased fatty acid oxidation and also increased apoptosis with lipotoxic cardiomyopathy. But I think the increased fatty acid oxidation I'm guessing at this point, based on the diagram, but when you had the massive increases in fatty acid oxidation from the saturated fatty acids there, you probably generated even more ROS that signaled apoptosis at the cellular level.
Right. Yeah. It's probably at that point a feed forward reaction. So, yeah, I mean, the important part here is when you have such excessive amounts of oxidative stress that you're decreasing SCD1, you're in like in this case, and if that's happening in the heart cell, you're in a lipotoxic cardiomyopathic state. Definitely not something that I would say you would want to be increasing. And so they have a quote that's discussing this. This is from that study where it says that increased expression of SCD1 might protect from palmitic acid-induced excessive fatty acid oxidation and apoptosis through mechanisms involving the downregulation of AMPK, inhibition of ceramide and DAG synthesis, and inhibition of mitochondrial ROS generation. So again, it's basically saying that SCD1 is a protective mechanism. This is on that left adaptive side. It's a protective mechanism to protect against excessive reactive oxygen species production, excessive fatty acid oxidation, and eventually excessive apoptosis. So, and then to continue on with that quote, it says, we suggest that this period is an adaptive stage. On the other hand, SCD1 expression is reduced when oxidative stress is increased. In this condition, saturated fatty acids are no longer converted to monounsaturated fatty acids, and ceramide synthesis increases, and as a consequence, strong oxidative stress contributes to the onset of lipotoxic cardiomyopathy. Thus, we propose this as a maladaptive stage. So, to clarify and relate this to Brad's point of view, he's suggesting that that's essentially what we want. We want to be in that state where you're producing so much reactive oxygen species from fatty acid oxidation that's happening, which A, I don't think is possible just from fatty acid oxidation, and B, I don't think is desirable.
Yeah. The other thing I want to point out here is they're using obesity and diabetes as the starting points here, and those states may be characterized by increased saturated fatty acid production by the liver, but they're also characterized by high amounts of endotoxin. So you don't only have saturated fatty acid causing the problem, you also have mitochondrial issues from the elevated endotoxin levels and usually I think it's gut-derived endotoxin. So you have like a chronic low-grade endotoxemia, and then you also have that generates the acute phase response, which increases saturated fatty acid production by the liver, but also increases a whole host of inflammatory mediators like tumor necrosis factor alpha, interferon gamma, and a whole and norepinephrine and angiotensin, et cetera, et cetera, et cetera, that has like a systemic effect beyond just, oh, saturated fatty acids are being produced by the liver and to, and push towards triglycerides or whatnot, what have you, whatever it is. So there's like, it's a systemic issue. It's not just a question. They're not just states of excess nutrient, or I guess the overnutrition is what people like to say. Like there's actually like impaired oxidation of the nutrition in combination with nutrient deficiencies and possibly low grade infections and et cetera, or overexposure to polyunsaturated fatty acids. Like there's a whole lot of issues going on. It's not just, oh, your liver's producing too much saturated fatty acid and you don't have enough SCD1 or whatever the argument could be based on this like one pathway.
Yeah, no, exactly. I mean, that's a huge point, which is, again, another thing that Brad is arguing against is fructose and insulin, because supposedly those increase SCD1, and as that study was showing, they do in that particular. I think that was mice, but that's what you're getting at is the problem in these states is not just you have fructose and the fructose goes to the liver and just becomes fat. That's not what's happening. That's not what's going to happen in a healthy human.
Especially when you change the fructose source from just granulated sugar or purified fructose added at a percentage of the diet to say like orange juice. If you look at any of the studies where you have like orange juice or grape juice or whatever it is, like 100% juice, and then you put them in high fat, high sugar feeding controlled studies with rats, they actually are protective, which shouldn't be explained based on their sugar content, which they're high in, obviously.
Mm-hmm. Including fructose.
Yeah. I mean, they're 50% fructose.
Right. Yeah. Yeah. And we have talked about that quite a bit. I'll link back to some of those episodes where we talked about why fructose is not a problem and why sugar is not a problem and why it doesn't inherently lead to fat accumulation in the liver. I'll link to a couple articles on that, where I've talked about that too. But to kind of summarize it here, you've touched on it a few times. We've kind of alluded to it, but basically, in a healthy human, we should be able to handle quite a bit of fructose in the liver. It gets converted to glycogen. Some of it is going to be used directly to produce energy, and some of it will be converted to other, converted to glucose, converted to other things, and then released out of the liver and kind of spread out throughout the body. So we're able to handle quite a bit of fructose when we're healthy, but there are a lot of things that can prevent that from happening, and instead will cause that fructose to become fat. One of those is endotoxin. Another one is polyunsaturated fats. Another one is if you become a rat, and that's because rats' livers are generally shown to be able to handle very small amounts of fructose compared to ours before they start converting it into fat, and that makes sense just because we need to have a much more capable liver, considering that our liver is basically a storage organ of fuel for our brains, of glucose for our brains. And so the fact that fructose goes directly to the liver, it's kind of that fructose ends up basically being that main fuel for our brains. It gets converted mostly to glucose and then glycogen in the liver, and then that ends up getting released for our brain to use. So considering that we have much greater brain capacity and demands compared to a rat, we need to have much better functioning livers that can handle a lot of fructose, and ours happen to do that. And so that's why you see rat and mice studies where when they have fructose, they tend to convert a lot of it to fat. There's also studies showing that even that doesn't even happen if you take away polyunsaturated fats, or if you block metabolites of the polyunsaturated fats, and or as you said, germ-free mice is another situation like that. So there are, even in rats, they may be able to handle fructose more than it seems to even show in the research if they're not exposed to endotoxin and polyunsaturated fat. And the problem is that in most of the fructose research, they're exposed to both of those things, especially endotoxin, because normally they're feeding pure fructose, which is not well absorbed and leads to endotoxin production in the gut. But even without the pure fructose, even if it's pure sucrose, that can still cause similar issues, and on from there.
Yeah. And I mean, the other thing is it's kind of rare to find isolated fructose in nature. I mean, yeah. I think the only real sources are if you had apple juice or like honey or agave nectar, which, I mean, usually it will come with glucose, and eating a lot of those the other thing too, I mean, eating a lot of those, you'll probably get digestive symptoms as well as whatever liver issue you're going to get. Because it'll induce, for some people, probably bloating and diarrhea. But then the other thing too is for rats, digestive physiology and anatomy, it is pretty different as well. So it's kind of like you can't really draw a parallel from, oh, I'm going to feed rats X grams of or X percentage of diet of free fructose. It just doesn't cross well. And then the other thing too is in a lot of like in some studies, even with humans, where they don't really show necessarily the same thing, but in overfeeding studies, like you're probably going to see increases in SCD1, and that's if you're going to overfeed somebody by 1,000 calories a day, their liver will probably increase fat production, and then will probably increase SCD1 to export the fat is in a monounsaturated form. So I mean, and that's expected, that's understood. Like that's what's supposed to happen if you're going to overfeed. And a lot of them, it's like in the overfeeding study, it'll be high amounts of, something in the form of like glucose or fructose or sugar and white flour and some type of specific type of fat to form a muffin or something like that. So again, it's still using purified nutrients as the sole source for the experiment.
Right. Well, and even in those overfeeding situations, normally they're limited by other factors like nutrient availability, polyunsaturated fats that are going to lead to more lipid accumulation. Like just eating an extra 500 calories or 1,000 calories doesn't even necessarily mean that that's going to happen. It depends on the context. But for example, when they looked at athletes who they're kind of assuming are metabolically healthy, it takes thousands of grams of carbs before you're seeing considerable amounts of lipid production in the liver.
Thousands?
Yeah, in that carbohydrate overfeeding study, they weren't really producing significant amounts of fat from carbohydrates until they had around 1,500 grams until they were taking about 1,500 grams of carbs over a couple of days. It wasn't until that second day of eating basically 800 grams of carbs that they were starting to produce some amount of lipids. So part of that was because they had to refill a lot of glycogen, but they were also oxidizing a huge, pounds of carbohydrates, like half a kilogram, which I guess is like a pound of carbs that was just being oxidized. So yeah, I mean, at that point, SCD1 would be regulated, as you were saying, but it does require quite a bit of overfeeding.
Yep. And I'm just saying it makes sense that SCD1 would upregulate with de novo lipogenesis at that point.
Yeah. And I also don't think that that's really a concern physiologically. We've talked about how our hunger signals at that point would be telling us not to continue eating, assuming that things are functioning well, you're oxidizing the carbs well, you have enough energy. That's going to turn off those hunger signals. I'll link to some of those studies. And when we've talked about that before, where basically ATP levels in the liver and brain are some of the main regulators of hunger and also allow for leptin to actually function properly, where if you have a lack of ATP, the leptin signal won't actually turn off hunger. So, and that's something. Go ahead.
Well, I was just going to say, I mean, assuming you're not eating Oreos as your main carb source, then you would probably be generating ATP from that carbohydrate, because it'd be coming, if it's coming from sweet potatoes or fruit juice or fruits or whatever, then within those categories, then it would be coming with nutrients as well to allow the carbs to be oxidized, which is also an important point, because
right.
And that this is one of the issues you see with some people with diabetes or obesity, whatnot, is you're eating a high amount of refined carb source. So what you see in the rat studies, and depending on, you can basically deplete B vitamins, which are necessary for the utilization of those carbs. So there's a lot more to the picture than just carb intake by itself. It's like what type of carbs you're eating. A lot of these rat studies, it's fructose, sucrose, glucose in free form, and that's what like it's just added as a percentage of the diet. Mm-hmm. Like the rat diet can literally be soybean oil, casein protein, some type of whatever your carb source is, whether it's starch, sucrose, fructose, or free glucose in isolated form, a vitamin mineral mixture, and then some methionine, because casein is slightly deficient in it. Right. So it's just like the most purified. So unless you're moving in that direction, which would be, I guess, akin to eating Oreos, which also has other stuff in there.
Yeah.
Like if we're talking about like a whole foods diet, it changes the picture pretty specifically. And your fruit juice, your whole fruit, your dried fruit, sweet potatoes, white potatoes, etc.
Yeah, assuming low endotoxin, low PUFA, all of those factors too. And one thing you were talking about earlier too is fructose absorption, and you had mentioned like apple juice and agave and things like that. And I did want to clarify also that some people don't have an issue. So I wanted to clarify first off what you're saying is those are all sources of carbohydrates that tend to contain more fructose than glucose. The ratio of fructose to glucose is higher.
A lot more fructose than glucose.
Yeah, but that isn't always an issue. Some people, are able to, even something as small as 5 grams more fructose is able to cause an issue, which could actually, depending on how different that ratio is, could require a decent amount of carbohydrates. It depends before you get to 5 grams more fructose than glucose. But for some people, they can absorb even a lot more free fructose than that. So there is individuality there.
The study I think I read on it was between 5 and 50 grams for participants with an average, I think, around 20.
Yeah.
If I remember correctly. Yeah.
So with that being said, some of those higher fructose foods might not be an issue for a lot of people, but that's kind of individual.
Yep. I agree.
Yeah. And also you were, again, in talking quote unquote negatively about those things, you do also have in those cases some of those beneficial plant compounds and nutrients that will help in other aspects of
especially
ensuring.
Apple juice.
Exactly. Yeah.
Yeah. Agave and honey are relatively, they have other components in them, but it's mostly just sugars in a solution form.
Right.
A combination of free sugars and sucrose.
Exactly. Yeah.
Yeah.
So circling back to SCD1 and Brad's views, one of his other points that we talked about was the polyunsaturated fat side, where the polyunsaturated fats do decrease SCD1, which he sees as a bad thing. But he also sees he sees as a good thing, but he sees that they supposedly decrease oxidative stress when they're burned in the mitochondria, which we would say is not necessarily accurate. We talked about that in the last episode, that they end up increasing oxidative stress because most of the time the concern is not what's happening when they're being oxidized in the mitochondria, but what's happening outside of that. If
they're incorporated into the actual mitochondrial membrane.
Yeah.
Also reaching the process of getting from your mouth to your mitochondria. Are they peroxidized in that process?
Right. But so I did want to point out some discrepancies here too, or just some issues that with the idea of PUFA being either harmful or beneficial, depending on whether their ability to decrease SCD1 is able to outweigh their negative effects when they're being oxidized in the mitochondria. And, I did think it was, so I'm going to include a couple of quotes of his, and then we'll, I guess, kind of discuss that because I don't want to be putting words in his mouth. So he says, all unsaturated fats lower SCD1 production, polyunsaturated fats more than monounsaturated fat. Bear fat can be relatively high in the polyunsaturated fat linoleic acid, similar to corn-fed lard. So if a native person is eating bear grease, SCD1 will be lowered to create an overall relatively saturated blend of body fat. The same thing is true of monounsaturated fats to a lesser degree, but there are a number of unsaturated fats that inhibit SCD1 much more specifically and to a far greater degree. So
there's a lot going on here.
Right.
With that statement.
Yeah. But the main point is that he's saying that at least the lowering of the SCD1 is a good thing, but he also then states that he's still not recommending PUFA consumption. In fact, that's one of his main things that he's against. And he explains a little bit of this later on, where he says, paradoxically, the soybean oil and starch eating American has a lowered overall pressure to upregulate SCD1 compared to the dairy eating person due to the SCD1 suppressing effect of polyunsaturated fat in soybean oil. However, the specific fatty downregulators of SCD1, which are CLA, palmitoleic acid, arachidonic acid, and others, are much more powerful downregulators of SCD1, and soybean oil doesn't have them. That's problem number one. Problem number two is that stored linoleic acid will start to cause a pathological inability to drive ROS production when cells switch from burning glucose to burning fat. A small amount of ROS tells the cell it's burning glucose, and SCD1 should be upregulated. So he's basically saying that there are two problems with PUFA, and specifically he's talking about soybean oil in this case, just like kind of what maybe a typical American diet would involve. He's saying, for one, yes, it increases SCD1, but not very strongly, basically not as strongly as CLA, palmitoleic acid, and arachidonic acid. although arachidonic acid would be a downstream metabolite of the linoleic acid he mentions. But
it'll be but it'll be in preformed version in the dairy.
right.
Because it'll just be, there's not linoleic acid, but there will be arachidonic, even in small amounts.
Right. And so then problem number 2 that he's saying is that when you're storing the linoleic acid, that's going to lead to an inability to produce enough reactive oxygen species when it's being burned
by the fatty tissue. So then you'll basically have insulin sensitive fat tissue is the argument.
Right. So yeah, I guess I don't have too much to add there. I just don't see it as,
I don't see this as like the foundational principle to focus on. Especially, it just doesn't.
You're saying like, SCD1 or
SCD1?
Yeah.
Like, it just don't see it as like the primary metabolic switch that we're looking for to determine what makes you obese or not. Because especially because it's just the mediator in like the chain, where basically anytime you have increased de novo lipogenesis or our influx of saturated fats into the liver is going to upregulate SCD1 to convert them to monounsaturated, some of them to monounsaturated fats to export them to other tissues throughout the body, because the body needs a certain amount of monounsaturated fats as well as saturated fats. I don't see it being the make or break on the entire situation, especially since we already broke apart the idea that you need to generate X amounts of ROS to shut down insulin sensitivity at the fat cells level. It just like that part of the theory just doesn't seem like it's going to hold water. So, and the other thing is the body seems to be upregulating SCD1 on purpose. Like it needs, apparently my guess would be that the body needs a certain amount of monounsaturated fatty acids. So when you have, trying to modulate around SCD1 is just sort of like beating around the bush of SCD1. Well, if we have enough PUFA, then we can lower SCD1. And it's just like, well, yeah, because you don't need to produce the monounsaturated fat now because you have a more unsaturated fat. But even from our argument, that's not necessary, because the polyunsaturated, if you're going to intake a lot more polyunsaturated fatty acids, and he already recognized it's not a good thing, but now you're going to have a lot more peroxidative damage, or you're more likely to have peroxidative damage with an increase in polyunsaturated fatty acids. So, and then for him, it was, they'll be stored in the fat tissue, and then they'll be insulin. They'll be more insulin sensitive. I just don't see where like this being the fulcrum or the main point to focus on in the system, especially when you start looking at confounders for endotoxin in a lot of these states as well. I think it's more likely that in those states, the endotoxemia is causing the issue and driving the increased deposition of fat by changing hormonal function and also mitochondrial function in general. And then upregulating liver fat production. I don't think that the SCD1 is causing the problem.
Yeah. I agree. And just kind of focusing in on the PUFA situation as well. We talked in the last episode about the problem being that, like, from our view, he's viewing these as agents of decreasing oxidative stress, and we view them as increasing oxidative stress. And I think that this is kind of similar here in terms of the SCD1 situation, where he's saying that the that the polyunsaturated fats are going to end up leading to more saturated fat basically being stored. And that may or may not be true in storage. It's relatively beside the point, I would say. But I mean, like, by that knowledge, you're also just going to have a huge accumulation of saturated fat in the liver. And anyway, the other side is that we know that the amount of polyunsaturated fats that are consumed is going to be reflected in the membrane composition of the mitochondria and of the cell as a whole, which, whether or not it's a membrane, again, doesn't really matter here. It's going to affect the structural components here. And that's going to directly lead to some of those issues that we talked about in the last episode, as far as increased lipid peroxidation, a very decreased ability to produce energy, basically a direct uncoupling effect, which he's saying is a good thing in the first place.
Yeah. So. It just doesn't, It's like making SCD1 the centerpiece and then trying to modulate around that just doesn't make sense to me, because your body is using SCD1 just when you have higher states of saturated fat in the liver to convert it to unsaturated. Like, I don't see that as a pathology. I don't see that as a problem. I don't see that as driving lipogenesis, especially. And I said this before, just like we don't have evidence that we're generating enough ROS with saturated fatty acids oxidizing in the fat cells to induce this insulin resistance at the fat cell level. Or that's not to induce the insulin resistance, but to turn off SCD1, excuse me, because we do know that those fat cells become more insulin resistant with saturated fat oxidation.
Right.
So I don't know. I'm not seeing that as a primary driver,
Right.
especially when the states you're comparing it in, where like we have an unknown and understood mechanism of why SCD1 is upregulated in states of obesity and diabetes and impaired glucose tolerance, fatty liver, whatever it is.
Yeah. I mean, I basically see it as another offshoot of the exact, the reactive oxygen species theory of obesity, which of course it is. I mean, that's why he's putting them both forth. They go hand in hand. It's the same, from my view, the same reductionist idea coming from hormesis. It's the same hormetic argument. And when you all end up arguing for that, you also end up arguing for basically anything that causes stress, which I don't think is always really recognized. But
it's like, let's cause a pathology so the body will have an adaptive response.
Right.
Like, so what happens if the pathology is not there?
Or what happens if it is there? Are you supposed to create further pathology?
Well, I mean, when we went through the graph, that's essentially what the point of view was.
Right.
It's like, we want to get to this point where we have this ridiculous amount of basically toxicity is what the argument seems like it's for. At least from my perspective, I don't want to put words in his mouth or create a straw man or anything. But it's like, we want to generate enough ROS to shut down SCD1, but it's already recognized in the article, I think that he actually posted, that shutting off SCD1 with extensive amount of ROS, number one, didn't seem like it was possible to be just from fatty acid oxidation, but also number two, it would actually cause apoptosis and fatty acid toxicity within the cell, lipotoxicity within the cell. So
right.
I just don't see I don't see this mechanism as being very relevant, as being like something to focus on for like excessive amounts of weight loss. Now, whether or not his dietary strategy works or not, as far as like focusing on saturated fatty acids and whatnot, is like, I don't disagree with him there, right, as trying to avoid PUFA in general. And keeping more towards the saturated fatty acid. But again, I don't see eating monounsaturated fatty acids as a problem either. I just don't think that this is the main mechanism of the issues, especially in states of obesity or diabetes or impaired glucose tolerance or non-alcoholic fatty liver disease. I see those as states characterized by high amounts of endotoxin, and then endotoxin inhibiting mitochondrial function, and then also driving an acute phase response, and then at the same time changing the hormonal profile to one that induces deposition by upregulating adrenal function and cortisol and whatnot. So that seems more of an important mediator than anything to do with this one enzyme in the liver and pushing high amounts of reactive oxygen species, which I don't even think is a good idea, especially through this mechanism.
Right. And I like that you mentioned apoptosis and that maladaptive side, because that's another one where, I don't know, you hear that all the time. And again, this is kind of coming back to hormesis, not necessarily something that Brad has said. But, apoptosis is a good thing. It's just, it's clearing out dead cells, and it's anything that stimulates it is going to be good, just like autophagy. And it's like, that's the problem with looking at these things in a reduced form, not in the greater context, not as a systemic whole, is that then you consider something like apoptosis as generally good, yet that's what you see in that study where they were showing lipotoxic cardiomyopathy.
Yeah.
So, and I think the SCD1 is an equivalent situation, focusing on that as the main determinant of whether something's healthy or not, is its effects on SCD1. And along with that too, I just wanted to circle back to one thing that I mentioned as far as hormesis goes, or you kind of mentioned too, which is just that when you're in a dysfunctional state, that's when you're seeing excessive amounts of oxidative stress. You're seeing dysfunction in adaptive stress pathways. You're seeing excessive fatty acid oxidation. We know that that's the backup form of respiration. And yet, if you're arguing in favor of hormesis, then what you're saying is that those dysfunctions are the product of a lack of oxidative stress, and you just need more. And yeah, I just think that, that study that we were talking about and with the SCD1 happened to be a really good example of showing that that's not the case. When you add oxidative stress on top of pathology, you end up with, like that quote, what they call
Just crippling of the
maladaptive.
cell structure.
Yeah, death, destruction.
Yeah, which is not what you want.
Right.
So, I mean, it comes back to what we talked about in the beginning. Like, while some of our views on like strategies or implementation in the real world, not on this more theoretical world, as far as, saturated fats are good in a diet, are, probably better than the unsaturated fats, where the focus on like either butter, beef tallow, chocolate, coconut oil, and even monounsaturated fats, which is, I guess, a discrepancy with him. Like, where I would say that, quality olive oil or macadamia nut oil wouldn't be a bad thing. I mean,
yeah,
I don't think that, like, I haven't seen them to cause massive amounts of weight gain, to be honest, especially considering I eat a decent amount of monounsaturated fat. I mean, N equals 1, but, and people that I've worked with using those oils hasn't caused the issue. But I feel like the pathway itself, or the idea, or like the lens that we're looking at it, this hormetic lens, is like, besides being like very focused and I guess a little bit reductionistic, it also, like we already went through, it just doesn't pan out. Like, we're not like, how could just oxidizing fats create that much ROS? We're not seeing it. And then, like, we're seeing protective effects of SCD1 in certain states. And it's like, in other states, yeah, when they didn't have SCD1, they didn't get diabetes or obesity or whatnot, but that was dependent on like the research model. Because I think before we did the last video, when we were going through some of the SCD1 stuff, when they fed the rats an increased amount of saturated fatty acids on top of not being SCD1 knockout mice, they actually developed issues where in the other models they had fed them high amounts of polyunsaturated fatty acids. And so, like, when you then exacerbate where the pathology is in terms of like feeding them high amounts of saturated fatty acids without SCD1, then they develop serious issues. And so the SCD1 is there for a reason. I mean, it's there to modulate any high, like, and again, this is, in just because saturated fats decrease or don't oxidize as much as polyunsaturated fats doesn't mean that then eat unlimited amounts of saturated fats and it won't cause an issue. There's still, the body has a preference for a certain amount, and it's regulating that by SCD1. So trying to dance around the SCD1, it doesn't seem to make, especially when we already have the context of the SCD1 issue being related to possibly being related to all these other factors, or like the obesity and diabetes and fatty liver being related to other contextual factors around seeing the upregulation in SCD1. Like, it's an association. And it's because in those states you have upregulated fatty acid synthesis in the liver. I don't know if I'm being very clear, but it's like, I see it as the SCD1 is an associated factor because in all these states you have upregulated fatty acid synthesis in the liver, which we already know is saturated. I see it more as that, and the context of why these states are developing being the actual issues, and not that the liver is producing fats and then SCD1's converting them to monounsaturated fats. I don't see that being an issue.
Yeah. And of course, if it's happening in excessive amounts, it's a sign of dysfunction.
Exactly. Yeah.
All right. That's going to wrap up this series discussing the Croissant diet. If you did enjoy it, please leave a like or comment if you're watching on YouTube. And if you're listening elsewhere, please leave a 5-star rating on iTunes or a review. All of those things really do a lot to help support the podcast and are very much appreciated. To check out the show notes for today's episode, head over to jfeldmanwellness.com/podcast, where you can take a look at the studies, articles, and anything else that we referenced throughout today's episode. And if you are dealing with any low energy symptoms, whether that is weight gain, chronic cravings and hunger, fatigue, chronic pain, brain fog, poor sleep, digestive symptoms, or hormonal imbalances, or any other sorts of low energy symptoms or chronic health conditions, then head over to jfeldmanwellness.com/energy, where you can sign up for a free energy balance mini course where I will walk you through the main things that you want to do to maximize your cellular energy, and I'll also explain how these symptoms and conditions really come down to a lack of cellular energy, and again, how you can adjust your diet and lifestyle to resolve them. So to sign up for that free energy balance mini course, head over to jfeldmanwellness.com/energy. And with that, I will see you in the next episode.